And we're back with part 2 of cells. We're picking up with the prokaryotic cell walls. Depicted here on this image are brick-like structures that are labeled NAG and NAM. These are modified carbohydrates. I'll show you on the next slide a little bit more detail regarding that. NAG and NAM subunits are linked together by peptide bridges. These little blue balls represent amino acids that are responsible for the linkage. This overall structure forms what's known as peptidoglycan. That's spelled out for you on the next slide. This is a bacterial cell wall, not an archaeal cell wall. This is the structure that's so important to bacteria. Almost all of the different species of bacteria have a cell wall. Of course, this is nature, so there's always some exceptions. Nature often breaks the rules. What do cell walls do? I know you're dying to know the answer to that question, so I'll tell you. They provide overall structure and shape. They do protect the cell as well from the forces of water, which may try to rush in specific environments. Bacteria come in different shapes. There's some common shapes, but overall their structure and shape is determined by how the peptidoglycan is linked together, and it's protecting the cells from osmotic forces. This is really important, especially when the cell finds itself in an environment where water is trying to move into the cell. The cell walls prevent the cell from breaking like an overinflated balloon. The cell wall, or the components that are linked to and embedded within the cell wall, can be very important for helping the cells attach to each other. It also helps them elude the effects of some antimicrobial drugs. Also, it may be important to note right now that since peptidoglycan is a molecule that differs from molecules in our own cells, thus it is an important target for many of the antimicrobials, the antibiotics that we use to treat infection. Remember too that bacteria and archaea have different types of components in their cell wall. Peptidoglycan is a bacterial structure. On this next slide, we're looking more closely at the chemical structure of peptidoglycan. NAG and NAM are shown, and they're compared to glucose so you can see the difference. Glucose is modified to produce NAG and NAM. Can you see the differences contained in these orange and blue colored areas of the NAG and NAM? Once again, this highlights that glucose is an important molecule for cells. It forms the basis of many other molecules used by the cell. Now let's look at the difference between the cell wall structures in gram-negative and gram-positive bacteria. They both have peptidoglycan-based cell walls, but they do have differences. Starting with the gram-positives, this is an image from your textbook. You'll see that the peptidoglycan shown in this peachy color is thick, and it's interspersed with some molecules called teichoic acid, or modifications of that called lipoteichoic acid. This molecule, teichoic acid, and its modifications are specific to gram-positive cells. Gram-negatives don't have this particular structure in their cell wall. Gram-positive bacterial cell wall is thick, which is different than gram-negatives, and it's outside the cell membrane. The cell membrane is depicted here below the cell wall. This is what's separating the cytoplasm from the outside world. In gram-positive bacteria, the cytoplasmic membrane is internal to the cell wall. The cell wall is the structure in closest contact with the environment. You can see that the cell membrane has these purple structures inside. These are different types of proteins, which we'll talk about shortly. So just to summarize, the gram-positive cell wall is relatively thick. It contains teichoic acids or lipoteichoic acid that are helping to anchor the cell wall to the cell membrane. And when you stain gram-positive cells in the gram stain, a staining procedure we'll talk about in several different times during the course, the cells will retain the crystal violet. Gram-positive cells will thus appear purple or violet after staining in the gram stain procedure. This is important because this is one of the very first procedures that you do in order to try to identify which bacteria you're dealing with. A modification of gram-positive cells are the bacteria in the acid-fast group. This is the group of bacteria that causes tuberculosis, as well as the leprosy-causing bacteria. The acid-fast bacteria have up to 60% mycolic acid in their cell wall, which creates a very waxy type of surface structure. This helps them survive desiccation or drying out. Acid-fast bacteria are a modification of gram-positive cells. They have one membrane and a thick, waxy cell wall. Gram-negative cells are a little more complex in structure compared to gram-positive cells. You can see that depicted up here, there is a cytoplasmic membrane, like the gram-positives. They have a thin cell wall. And then they have an outer membrane. Gram-negative cell walls are located in between the two membranes, and it's relatively thin compared to the cell wall of gram-positive bacteria. In the outer membrane, they have a unique component called LPS, or lipopolysaccharide, a molecule that will be highlighted often during this course. The LPS molecule is extremely toxic and a potent activator of our immune system. Therefore, it also goes by the designation endotoxin. It's something that really gets our immune system ramped up. LPS is a component of the outer membrane of gram-negatives. Gram-positives do not have an outer membrane, nor do they have LPS. When you stain gram-negatives in the gram stain, they do not retain the purple dye, and they will appear pink when you finish the gram stain. The gram stain will be described in even more detail coming up. Something important to keep in mind is that there are frequently exceptions to the rules humans use to describe living organisms. As you might imagine, there are exceptions for cell wall structures in prokaryotic cells. There are, in fact, bacteria without cell walls. The bacteria without cell walls belong to a group that might cause an infection called walking pneumonia, as well as other problems. We will discuss specific infections towards the end of the semester, but it's important to note here that mycoplasma a genus of bacteria, do not have cell walls. These bacteria tend to be smaller than other typical bacteria. They can squish through small pores or holes because since they don't have a cell wall that is making them retain a specific shape, they can almost flow through a small opening. Mycoplasma do have other features of prokaryotes. They're definitely cellular, definitely bacteria. I would like to point out that this genus Mycoplasma is not the same as the genus of acid-fast bacteria, which were Mycobacterium. The first part of the name is the same, so if you weren't paying close attention, you could mix those up. Mycoplasma are a cell wall lacking genus, which is an exception to the general structure of the bacteria group. Now a quick word on archaea. They don't have peptidoglycan. They have other types of polysaccharides or proteins that make up their cell wall. They do have gram-positive and gram-negative types of structures that stain in a similar way that the bacteria do in a gram stain, but they are a bit different, chemically different cell wall structures. Moving to cell membrane. Of course, the cytoplasmic membrane is an important structure because it separates the internal environment of the cell from the external environment of the cell. The analogy that is used to describe the cell membrane is fluid mosaic. What does that really mean? Let's break it down. Fluid. Something fluid flows like a liquid. The mosaic term is referring to the fact there's a lot of different components that exist in the membrane. Together, these parts form the larger picture, just like an artwork mosaic. I like to conjure an image of the surface of a swimming pool with many toys floating in it. The surface of the pool is the membrane, and the toys are the protein and other components. The components can float around the surface. This is similar to the components in the lipid bilayer of the membrane. The phospholipid bilayer is comprised of those phospholipids with the fatty acid tail ends facing each other. Remember what we talked about in the chemistry section in the last chapter? The fatty acid tails are facing each other. They're hydrophobic. The head groups, the phosphate groups, are facing the outside. They like the water. They're hydrophilic. This is important because the cytoplasm of the cell is mostly aqueous, as is the external environment of a cell. So the water-loving components, the hydrophilic phosphate head groups on the phospholipids, face the outside world. In the image, the purplish components are the proteins. Notice that the proteins can span the membrane, meaning both ends of the protein face external and internal cell environments. This means that the protein itself has hydrophilic regions as well as hydrophobic regions, so it can exist within the membrane and into the environment. The hydrophobic and hydrophilic regions are determined by the amino acids that exist in those regions. These proteins fold in a specific way to give the protein this overall structure so that it can embed itself in the membrane. These membrane proteins can form channels, or they will bind something on the outside of the cell and trigger a change in shape that will trigger something to happen on the inside of the cell. These proteins that span and embed the membrane are termed integral proteins because they are a part of the membrane structure itself and because they span the membrane. Integral membrane proteins are key in helping the cell function. Some proteins do not span the membrane but are more loosely affiliated with the inside of the lipid bilayer. These are called peripheral proteins. While they're not spanning the membrane, they do attach or are attracted to the edges of other molecules that do span the membrane because they often play a role in communicating signals coming from integral proteins. When they detect a shape change, a conformational change, or a chemical activity of the integral proteins, they then will perform a chemical reaction to begin some sort of activity inside the cell. These membrane proteins depicted here play different roles. They could be receptors, they can be carriers or channels to move things through the membrane. They may perform a chemical reaction to propagate a signal, therefore acting as enzymes. Remember, the integral proteins span the membranes. The peripheral proteins are loosely attached to the inside. Glycoproteins have a sugar, the glyco component, linked to it. The linkage was made after the protein was produced, an enzyme attached to sugar group. These glycoproteins are typically located on the surface of the cell and often proteins that talk to other cells or signal other cells or form some sort of receptor. This next image from your book is depicting a protein that's integral to the membrane and is forming a channel. These proteins play a starring role in helping the cell create gradients of charges. We'll talk about hydrogen ion gradients in chapter 5. A hydrogen ion gradient is depicted here. Notice there's a lot more hydrogen on the outside of the cell compared to the inside. This gradient, this ion concentration difference, is key for producing energy for the cell. By creating these gradients, a cell can use it to produce ATP. More on that in chapter 5. In photosynthetic prokaryotes, the cell is able to harvest light energy with components that are within the cell membrane. This too is detailed in chapter 5. The membranes are said to be selectively permeable. In a simplified explanation, this refers to the fact that not everything can cross the membrane. Only certain molecules are allowed to cross. That makes it selectively permeable. The protein channels tend to dictate what can cross the membrane and in what direction. That can be a passive or an active process, which we'll come to in a minute. So the membrane is maintaining this concentration and electrical gradient within the cell in order to allow the cell to function. Chemicals, molecules, are going to be concentrated on one side or the other, and that actually is going to create a voltage as is shown in this image. Let's take a look at the processes directed by these cellular proteins in the membrane, the gatekeepers, which allow things to cross. First, we'll examine the passive processes, meaning the movement of molecules across the membrane that does not require energy. Crossing the membrane without an input of energy might entail simple diffusion or facilitated diffusion. This slide is showing us the different scenarios for this passive type of diffusion. We can have a molecule that is the right chemical composition and small enough that it might actually pass through the membrane. These tend to be very small and hydrophobic types of molecules. They can't have a charge. They're not polar, and therefore they don't really dissolve very well in water, but there'll be some within the aqueous environment, and those small hydrophobic molecules can cross the membrane because they're not impeded by the hydrophobic barrier of the fatty acid tails. Only a few types of molecules can do this. Steroid hormones are one type that can cross the membrane itself. That's diffusion. Facilitated diffusion requires a channel that will allow certain molecules to cross. It might let several different types of molecules cross, as shown in this image, when we see different colored dots moving across. We can have a more specific type of facilitated diffusion where only one type of molecule can get across, but these are similar types of scenarios. Now we can talk about water. A lot of times students have the misunderstanding that water just crosses the membrane outright. Water crosses the membrane very little through diffusion alone. The polar nature of water prevents its easy passage across the hydrophobic middle of the membrane. Only a small number of water molecules cross that barrier without some help. Living cells have what are called aquaporins. These are pores produced by an integral protein that lets water through, hence the aqua. Porin refers to a pore or hole. So these aquaporins are holes that let water travel either way across the membrane. This experiment is depicting what happens with a semi-permeable membrane, like the cell membrane. To do this experiment, you put a semi-permeable membrane, which mimics a cell membrane, here in the center. Then you fill this U-shaped tube with two different solutions. One has more dissolved stuff in it, and the other less. This stuff is more properly called solutes. The water is the solvent. Thus, solutes are the dissolved components in a solvent. In this case, in life, the solvent is always water. On this side of the tube, we have more dissolved solutes. On the other side, we have less dissolved solutes. We're going to give some terms to these solution situations. When we have solutions with two different concentrations, then we have a hypertonic and a hypotonic solution. But what we really want to depict in this particular experiment is that water wants to move to the side where there's a lesser concentration of water, because there's more dissolved stuff, more solutes. The water concentration would be lower compared to the solution where there is less solutes in the water. Thus, water is going to move towards the side with more solutes. The solutes can't cross the semi-permeable membrane, but the water does. So you might imagine these solutes could be proteins, salts, sugars, whatever you want the solute to be. Water is going to move across the membrane to try to equilibrate so that there will be equal concentrations of water on either side of the membrane. In this experiment, when water moves across, the volume increases on the one side of the membrane. So we end up with less volume on the side, this side, because water has traveled across the membrane to try to equal out the concentration of water on both sides of the membrane. All right, we're going to stop at this point and pick up on the next video with this idea. Part 3 coming up next.